Okay, thank you very much. We've heard a little bit about nucleoside analogs, so that's going to help me with some of the background of what I'm going to be talking about. As you are all aware, nucleoside analogs such as sofosbuvir have become one of the latest classes of compounds to receive approval for clinical use against Hep C. Compounds such as sofosbuvir are administered as prodrugs that must be intracellularly activated to their nucleoside triphosphate form before they can exert their antiviral activity. So in other words, the prodrug, such as sofosbuvir, must enter the cell, and the prodrug group shown in blue here, in green, has to be removed through intracellular metabolism, and then 3 phosphates have to be added through host kinases in order to generate a nucleoside triphosphate, or RNTP. And it is only this RNTP form that can go on and be a substrate for the viral polymerase called NS5V. So because this is a structural mimic of natural nucleotides, it'll be incorporated by the viral RNA into the growing viral RNA chain and cause chain termination. In this way, viral RNA synthesis cannot be completed, therefore the virus cannot replicate. In our laboratory at Emory University in Atlanta, we're very interested in developing new nucleoside analogs, and today I want to take you over some of our preliminary data with one of our prototype compounds, which I'll call 4-NOH cytidine prodrug. So the prodrug group of this compound is very similar to sofosbuvir. It is highlighted here in gray. This compound also has a chemical modification at the 4 position of the pyrimidine base, as shown in the red circle, and it also has the canonical 2'-methyl modification that Dr. Gota referred to earlier, and this is the chemical modification that's important for chain termination. So every time we have a new prodrug, the first thing we do is we look at its antiviral activity. In our in our replicon system, we observed that this prodrug had pan-genotypic submicromolar activity against all the genotypes tested to date. In the different cell lines that we tested, we haven't seen toxicity yet, up to 100 micromolar. We also haven't observed mitochondrial DNA toxicity. So this looked like a promising compound to go ahead with further analysis. So another postdoc in our laboratory looks at the intracellular metabolism of these prodrugs, and I want to share some of her data with you. So she did an egress study where she incubated 10 micromolar of the prodrug in HUH7 cells. After 12 hours, she washed away the prodrug, and then she used mass spectrometry to look at the metabolism of this prodrug inside the cells. So as you can see in the purple, at about 8 hours, we have a peak of total nucleoside triphosphate, or the potentially active metabolite, at about 8 hours. But what she also observed was that instead of just having one kind of triphosphate analog, she had 3 different species, all of which peaked at different times and had different half-lives. So it appeared that the metabolism of this prodrug led to a cascade of RNTP generation. So to reiterate this point, so we administer one prodrug, and as we expected, the prodrug group gets removed. We have a monophosphate that becomes a diphosphate, and then eventually we have a 2'-methyl 4-NOH CTP triphosphate formed right here. What was somewhat unexpected is that some of these intermediate steps can be converted at the base moiety here to a cytidine, so then you can have 2'-methyl cytidine monophosphate, diphosphate, and a 2'-methyl CTP generated. And similarly, these compounds can undergo deamination, and this generates 2'-methyl UTP. So the take-home message of this busy slide is that one prodrug is generating 3 different triphosphates intracellularly. So what are these triphosphates? 2 of them, 2'-methyl CTP and UTP, are known inhibitors of hepatitis C. They exist in the literature, and we independently confirmed by synthesizing these compounds separately and testing them in a cell-free assay with the purified enzyme that indeed they are active, and their in vitro activity is comparable to the active metabolite of sofosbuvir in our assay conditions. But really, the third nucleoside triphosphate, 2'-methyl 4-NOH CTP, hasn't been studied in the context of HCV before. So the rest of the talk will focus on the biochemical characterization of this metabolite. So 2 questions that we want to answer for the rest of the talk are, 1, is this chemically modified compound still incorporated by the viral polymerase? So is it a substrate? And if so, can it inhibit viral RNA synthesis? So the system that I'll be showing for the rest of the talk is fairly simplified. Here in gray, you have the purified NS5V RNA polymerase that is incubated with a synthetic 20-mer RNA template, and an elongation complex is formed, which allows for the specific incorporation of a CTP or a CTP analog at position 11, highlighted in pink. So when we incubate increasing amounts of either the inhibitor 2'-methyl CTP or the candidate TP metabolite for about 30 minutes, and then visualize that result on a denaturing polyacrylamide gel, what we notice is that, as expected, our control, when we increase the concentrations, yes, we see the appearance of this +11 band, which suggests it is a substrate. That's what we expected. But for the 2'-methyl-4-NOH, we also see a similar pattern, although it migrates slightly differently. So this suggests that indeed the third metabolite of this prodrug can be a substrate for the polymerase under these specific conditions. So the next question is, well, can it inhibit RNA extension? And here it's a very similar theme in terms of the experimental setup. The only difference is that we incubate increasing amount of the inhibitor at the same time as natural nucleotides. Okay, so the natural nucleotides are competing with our inhibitor. And when we have an inhibitor that works, shown on the right panel here, such as 2'-methyl UTP, the increasing amounts of the inhibitor should correlate with a decreasing amount of the full-length RNA formation, as you can see here. And this at the same time correlates with the appearance of these pausing sites along sites of UTP incorporation. Okay, so that's how we know that we have an inhibitor that's working in our hands and we can obtain an IC50 value. Now, if you look at the left panel with the 2'-methyl-4-NOH CTP, you can see that increasing the concentration of inhibitor does not correlate with inhibition of full-length RNA synthesis, and furthermore, we don't see any cytidine analog-specific pausing sites. So this suggests that under these conditions, when the nucleotides compete with the inhibitor, we don't see this inhibition. So of the 3 nucleotides generated, likely only 2 of them contribute to inhibition. So as my final results slide, we wanted to sort of address that discrepancy, that why is it that in one assay we saw that 2'-methyl-4-NOH CTP is a substrate, but then why can it inhibit RNA polymerization? So here we looked at, we applied enzyme kinetics experiments to look at the catalytic efficiency of this substrate, where, again, we incubate the elongation complex with varying amounts of the inhibitor or a metabolite, and we looked at this incorporation over time. In this way, we can obtain several different important kinetic values, and we don't need to go into the details too much. I'll just highlight. So by varying the time, we can obtain a rate of incorporation, so how well the compound is incorporated. So the higher this value, the better. We can also obtain a KD value or dissociation constant, which would say how well the compound is binding to the active site. So the lower that value, the better. And when we combine them together, the ratio of K-Pol KD is defined as the catalytic efficiency, so how good of a substrate we have. So if we compare the natural substrate CTP to 2'-methyl CTP, so the compound with one chemical modification, and similarly the UTP natural substrate to 2'-methyl UTP, we see that the addition of that methyl group causes about a 10-fold reduction in the catalytic efficiency, and that's expected. But with the 2'-methyl-4-NOH CTP, this catalytic efficiency value is reduced by 3 orders of magnitude. And that probably explains why, when the natural nucleotides are present, we don't see this inhibition. And probably what we can conclude is having a simultaneous chemical modification on the base moiety and the sugar moiety makes this a suboptimal substrate for the enzyme. And this is consistent with the inhibitory profile that we see. So to conclude, what I've shown you and summarize what I've shown you, 4-NOH cytidine prodrug represents a pan-genotypic, submicromolar, and selective anti-HCV compound. The metabolism of this prodrug generates 3 different nucleoside triphosphate metabolites. 2 of them appear to contribute to inhibition. 1 of them seems to be catalytically deficient. Nevertheless, we are moving forward with further developing this compound. I believe animal studies are next. And on that note, I'd like to thank my supervisor, Dr. Shanazi, and the members of our laboratory, our collaborators at Co-Crystal Pharma, and the NCRTP program, and my co-mentor, Rodney Russell, for giving me the opportunity to share our data with you. And I'll take your questions.
Hi, Maryam. Very nice. I was wondering about the resistance profile of your drug versus Sovaldi.
Yeah, versus Sovaldi. So in the replicon system, we haven't been able to select for resistance yet. In the JFH1 system, we're collaborating with the Russell Laboratory to see if we can select for anything in vitro. So comparing the compound to sofosbuvir triphosphate directly in the cell-free assays, we do see resistance to S282T with each of the metabolites separate. I've also tested some of the mutations that Dr. Gota mentioned, L159F, B321A. We don't see resistance, but we also don't see resistance with sofosbuvir. So it seems to be very similar.
I presume you have looked at the effect on other polymerases?
So yes, the 2' methyl compounds are substrates for example, purified dengue polymerase, but that is not necessarily true that the prodrug will be active against those polymerases. So when we tested the prodrug against vaccinia virus, dengue virus, influenza, we didn't see activity, but that might have to do with intracellular metabolism. But the active metabolite itself, I know at least against dengue it is active.